BZYCT 135. Bachelor of Science General BSCG. Term End Examination. June, 2024. BZYCT 135, Physiology and Biochemistry. Time, 2 hours, maximum marks, 50. One. A. Fill in the blanks with suitable words. I. Neutrophil is a type of leukocyte with multi-lobed nucleus and phagocytic in function. E. Angiotensin I enzyme that regulates blood pressure is secreted by kidney. E. Sarcomere is the functional unit of muscle fiber. IV. The deficiency of dopamine neurotransmitter causes uncontrolled muscle contraction in Parkinson's disease. V. The marine environment is isotonic to invertebrates such occupying that habitat. Explanation. I. Neutrophil is a type of white blood cell with a multi-lobed nucleus and performs phagocytosis, helping fight infections. E. Kidney secretes the enzyme renin, which leads to the formation of angiotensin I, important in regulating blood pressure. E. Sarcomere is the smallest contractile unit in a muscle fiber, located between two Z lines. IV. Dopamine deficiency leads to Parkinson's disease, characterized by muscle rigidity and tremors. V. Marine environments are isotonic to sponges, allowing them to maintain osmotic balance with their surroundings. B. Match the following. I. Trypsin 5 pancreas. E. Superoxide ion 3 free radical. E. Hexokinase 4 glucose. IV. Hemorrhagic factor 1 vitamin K. V. Glycogenesis 2 liver. Trypsin, a proteolytic enzyme, is secreted by the pancreas. Superoxide ion is a reactive free radical, damaging to cells. Hexokinase catalyzes the phosphorylation of glucose in glycolysis. Hemorrhagic factor refers to vitamin K, essential for blood clotting. Glycogenesis, the synthesis of glycogen from glucose, occurs in the liver. 2. Short notes. I. Exchange of respiratory gases in the alveoli. Brief, gas exchange occurs via diffusion between alveolar air and blood. Elaborated, oxygen from the alveoli diffuses into pulmonary capillaries while CO diffuses out into the alveoli due to concentration gradients. This is facilitated by the thin alveolar walls and moist surface. E. Pheromones. Brief, chemical signals that influence behavior or physiology of other members of the same species. Elaborated, pheromones are secreted by organisms to communicate, such as attracting mates, marking territory, or signaling danger. They play a vital role in reproductive and social behavior. Q3. Differences. I. RBCs versus WBCs. RBCs are red blood cells, biconcave, and lack nuclei. They contain hemoglobin to transport oxygen. WBCs are white blood cells and have nuclei. They defend the body against infections. RBCs are more in number than WBCs. WBCs are classified as granulocytes and agranulocytes. E. Membrane potential versus action potential. Membrane potential is the resting electrical charge across a cell membrane. It is usually around -70 mV. Action potential is a rapid, temporary change in membrane potential. It occurs during nerve impulse transmission. Depolarization and repolarization occur in action potential. Membrane potential is static, while action potential is dynamic. E. Aerobic versus anaerobic respiration. Aerobic respiration occurs in the presence of oxygen. Glucose is completely oxidized into CO and HO. It produces about 36 to 38 ATP molecules. Anaerobic respiration occurs without oxygen. It results in lactate in animals or ethanol in yeast. It produces only 2 ATP molecules. IV. Molluscan kidney versus Malpighian tubules. Molluscan kidneys are found in mollusks. They are metanephridial and filter body fluids. Malpighian tubules are present in insects. They excrete waste into the alimentary canal. Molluscan kidney filters coulombic fluid. Malpighian tubules absorb solutes and water from hemolymph. Q4. Digestive enzymes and carbohydrate digestion. Four major classes of digestive enzymes. 1. Proteases, digest proteins into amino acids. 2. Amylases, break down carbohydrates into sugars. 3. Lipases, break down fats into fatty acids and glycerol. 4. Nucleases, digest nucleic acids into nucleotides. Digestion of carbohydrates begins in the mouth with salivary amylase. Amylase converts starch to maltose. In the stomach, amylase is inactivated by acid. In the small intestine, pancreatic amylase continues digestion. Maltose, lactose, and sucrose are broken down by specific enzymes. Maltase converts maltose to glucose. Lactase converts lactose to glucose and galactose. Sucrase converts sucrose to glucose and fructose. Monosaccharides are absorbed in the small intestine. Absorption occurs via active and passive transport mechanisms. Q5. Molecular basis of muscle movement. Muscles are made of actin and myosin filaments. A sarcomere is the functional unit of muscle contraction. Myosin heads attach to actin to form cross bridges. ATP binds to myosin, causing detachment from actin. Hydrolysis of ATP energizes the myosin head. Myosin head pivots and pulls the actin filament power stroke. New ATP binds to release the myosin head. Calcium ions regulate this process. Calcium binds to troponin, shifting tropomyosin. This exposes actin's binding sites. Repeated cycles lead to muscle contraction. Relaxation occurs when calcium is reabsorbed. Q6. Enzyme regulation. Allosteric enzymes. These are enzymes regulated by molecules binding at a site other than the active site. The binding changes the enzyme's shape and activity. Allosteric regulation can activate or inhibit enzyme function. They often control metabolic pathways through feedback inhibition. Enzyme inhibition, competitive inhibition. Inhibitor resembles the substrate and binds to the active site. Prevents the actual substrate from binding. It is reversible by increasing substrate concentration. Example: malonate inhibits succinate dehydrogenase. Competitive inhibitors increase Km Michaelis constant but Vmax remains unchanged. Q7. Vitamins and enzyme reaction. 5 water-soluble vitamins and their deficiency diseases. 1. Vitamin B1 thiamine deficiency causes beriberi. 2. Vitamin B2 riboflavin deficiency causes ariboflavinosis. 3. Vitamin B3 niacin deficiency causes pellagra. 4. Vitamin B12 cobalamin deficiency causes pernicious anemia. 5. Vitamin C ascorbic acid deficiency causes scurvy. These vitamins are not stored and must be taken regularly. They support metabolism, immunity, and nerve function. Glutathione peroxidase (GPX) reaction. GPX is an enzyme that reduces hydrogen peroxide (HO). It uses reduced glutathione (GSH) as a substrate. The reaction is 2-GSH plus HO right pointing arrow GSSG plus 2HO. It protects cells from oxidative stress. GPX plays a key role in cellular antioxidant defense. It also reduces organic hydroperoxides in lipid membrane.